Good evening, everyone. So today the popular science talk is going to be delivered by Dr. Chitra Pattabiraman. Dr. Chitra Pattabiraman is a virologist or molecular biologist and uses genomic tools to identify pathogens, particularly in brain infections. She's the founder and chief scientific officer at Infectious Disease Research Foundation, a not-for-profit organization that carries out research at the interface of emerging infections in public health in India. Dr. Pattabiraman obtained her B.Sc in microbiology from Calcutta University, an integrated MSc PhD in life sciences from NCBS Bangalore, and was awarded a Serb Royal Society Newton International Fellowship for postdoctoral work with the Brain Infections Group at the University of Liverpool, followed by the India Alliance Early Career Fellowship carried out at NIMHANS, Bangalore. For the last 3 years, she's been involved in tracking the introduction, spread, and emergence of variants of SARS-CoV-2 in Karnataka and developing tools to visualize and analyze this data. So today she'll be talking about integrating genomics into outbreak response, where using examples, she'll explain how knowing the genomic sequence of pathogens can help us understand where it came from, how it's spreading and changing. So it's going to be a very enriching talk. And if you guys have any questions, you can ask her. We'll also have a question and answer session at the end of this. Over to ma'am.
Hi, good afternoon, everyone. I guess you can hear me. So I guess when physicists and statisticians and mathematicians want entertainment, they call the biologists. So yeah, that's what I am for this 1 hour. So we'll be talking about, so I'm going to talk to you about infectious diseases. I know everyone is a bit tired. How many of you feel COVID fatigue, like don't want to hear about COVID anymore? Okay. Okay, so I will talk about COVID a little bit, but I'll try to restrict it to how the virus was evolving, okay? And so this was how I used to begin my talk before the pandemic, with the idea that as long as humans have been around, pathogens that infect human beings have also been around. And we have been carrying that. So basically, since humans started living collectively, farming and things like that, we know that there have been viruses, there have been outbreaks, and people have carried these infections from one continent to the other, either via travel or conquests. And you can see that. So this is a person, sorry. This here is a person with polio in one of the Egyptian hieroglyphics. And this is a very old book called the Florentine Codex, and you can see people infected with smallpox. So there was like an entire like outbreak of smallpox that happened when people from Europe went to South America, and a lot of people who used to live there died out. This is from the plague, and what it basically tells you that outbreaks have been part of our historical narrative and what we became as a human race, as we it has gone, the history of that part of biology has gone in parallel with human beings and what we have done and wars and so on. And so very much in popular imagination. That's the flu. 1 in 3 humans across the world was infected. And this was around the time of World War I. So returning soldiers then brought back this virus from wherever they were fighting. And so there was local spread after that, and it killed about 50 million people. And this was, I think, one of those, a lot of lessons learned from that time, I think were helpful in understanding what can happen. So this was our best example of what a pandemic would look like before SARS-CoV-2. We now live in a very connected world, and I don't need to give this slide as an example anymore, but even before this pandemic, we knew that a virus that emerged anywhere in the world would get to other parts of the world really fast. So what you're looking at here is air routes, I think, with very specific airports. So this is not all the connections in the world, but this is some of the connections highlighted. And it already tells you that the world is very, very connected. And these are all the shipping routes, okay? So even if we are not human beings just traveling, things are traveling all across the world. and mosquitoes travel in some of these containers and so on. So there is some evidence that you do get spread of some diseases in this way. And so we have what we are looking at is a risk map. So what they've done here is they've tried to map out regions in the world that are at risk for the emergence of a new pathogen. Okay, and what did they use to predict these hotspots? The idea was that if you have a lot of so we are looking here, we are looking at zoonotic pathogens, and I'll tell you in 1 minute why we think that that's important. Basically, these are pathogens that come from animals into humans. Okay, so these are what we call spillover events, and you get the pathogen circulating in wildlife already, and then you get an introduction into humans. So these are called zoonotic pathogens, and this is where we believe that there is a high risk of something spreading fast in humans. And what you're looking at here is a sort of risk map, which is sort of predictive. This is based on things like the number of people living in that region, the amount of biodiversity, what is the interaction between the human population and the wildlife. So in areas where there is a lot of interaction between wildlife and the human population, and there's encroachment into where wild animals were usually living, for example, in South America, in parts of Northeast India and so on, you will find that the color is darker. We don't know if this is true, right? Because this is just based on our ideas of where we think pathogens emerge based on data for where outbreaks have happened. So we are only looking at outbreaks in humans, okay? We call something an outbreak usually when it gets detected and a bunch of humans fall sick or die. Usually it gets picked up when there are human cases and human deaths. But what we think is happening is that there are pathogens or viruses. Look at the time scale of this graph, it's about 3 months. We think that there are some viruses that are circulating in the wild population. Why do you think the curve looks like this? One explanation, does somebody have a thought on why the initial curve looks like this? It could be seasons. It could also be presence of susceptible individuals. So seasons, not so much because of the timeline here, but the pattern could be a seasonal pattern. But here it could be that it's all the young ones are getting infected or something like that. But still the timeline doesn't make sense. So it just suggests the amount of susceptible individuals present. Initially you have some who are susceptible, then they recover, and in that population there has to be something new happening here. So this graph, I don't know whether it makes sense in the background, but the idea is that there are some infected individuals in the wild population, but there is never an outbreak in that population. So it's sort of a reservoir species. And then there are these spillover events into possibly other animals, okay, first. And these animals are the ones that are in contact with human beings. And from these animals, you get an amplification in the animals. And then because the amount of the virus in the environment is more now, there is a chance that you will have a spillover event to humans. There is almost no virus that has been detected here in the first spillover. We have gone back and traced some of these events based on the number of mutations accumulated and so on. But I don't think anybody, these are hard to record and hard to figure out. You need to find animals that are sick, right? And usually you find humans who are sick. And here the outbreak, basically once the susceptible individuals are no longer there, it comes down. And if these individuals come into contact with other people and so on, you can get more than local spread. So before a couple of years, things like Ebola used to have very local outbreaks. So there weren't that many people. There would be small outbreaks in very localized regions. You wouldn't get spread outside of that. But now we are seeing more spread as well because people are traveling more. There are more contacts. And there is for some viruses, we think they also change so that the transmission may increase and so on. For example, for SARS-CoV-2, Omicron is way more transmissible than the initial SARS-CoV-2 that was there. So more people get infected. And these are the examples of some viruses that have emerged from zoonotic events that we know of. SARS-CoV-2 is also one of them. So what are the components of an outbreak response? And this is something that I have made up. I'm happy to talk more about it, but I think these are the rough components, and we can maybe add bits The first is that, of course, you have affected people. It's not just people, right? I just said that there is wildlife, there's domestic animals and so on. But let's say there's the affected population. Then there is this entire cycle of people who collect the information and all the research that happens, all of the epidemiology that happens in figuring out about who got infected, where they are, what is this bug. All of these things happen in this sort of research information gathering. And there are many, many agencies, many different groups of people who do this kind of work, right? And then this information gets sent to people who can make decisions at the policy level, or who can actually decide what to do. And this, as you can imagine, can happen at a very local level, right? So suppose you have an outbreak in a village. It could be the people who are making decisions at that level, or at the scale of a country or a city. So in each case, the stakeholders are going to be different. And the kind of information that they will need or they can act on is also going to be different. And the kind of implementation measures that are available to them is also going to be different. So the scales of this is different, but roughly the components of the system, I suspect, are going to be similar, where you have an information gathering system, and that has to somehow translate usually by people digesting that information and inferring something from it to some policies or implementation or interventions of what should be done. And this is how you impact the people, right? So the information directly, usually you need to go through some steps in order to make these changes. And so usually we are sitting here as researchers, as people trying to figure out how a virus is spreading in the population and so on. This is our place. And during the pandemic, this pandemic particularly, we've had to talk to people here and here in order to try and understand how best we can collect that information and analyze it. I'm happy to talk more about that. So suppose a new virus comes into town, right? What are the tools that we have in order to like figure out what it is? Do you know what the first one would be? Can someone shout out? Yeah, looking under the microscope. Yeah, so when do you think that's going to be useful? It'll be useful if it is something that can be seen, right? So you have the right microscope to look at it. Secondly, if there's enough of it in the sample that you're looking at, okay? So if it's, for example, for TB, many of the samples may not contain the bacteria. Okay, so that's why, and you may miss something like that. If you don't collect the right sample at the right time, you may not see it if there's not enough of it. So the biggest challenge with microscopy is having enough of the bug in the sample for you to see it. And what's the next one here? That's growing the bug. What do you think the challenges will be for this? Yeah, that's right. So we don't know how to grow many of these microorganisms. We don't know what would be the right conditions to grow them. So this kind of enrichment, which would make identification visually easy, is usually quite hard to do. Some organisms can be really slow growing. Again, TB needs very special conditions to grow, takes a long time to grow. You may not have that much time. Okay, if you wait 2 weeks for a culture to turn positive and someone is sick and you have an outbreak going in the community, this is probably not the best approach, right? But it has remained the gold standard. So if you ask people who do microbiology in an outbreak, what is it that they would do if they collected samples? They would try to grow it. Okay, so this has, but in the case of SARS-CoV-2, we didn't wait that long. And I'll tell you that story in 1 second. Host defense response, what does that mean? Has anybody done a test for dengue? Have you given a blood sample for dengue? Have you seen this thing? It will usually say IgM, IgG. Yeah, so IgM, IgG are actually looking at your immune responses to dengue. Okay, so if you have an acute response, that suggests that you had an infection recently. So IgM suggests a more acute, you had an infection. IgG suggests pre-existing, that is, you had memory to that infection. So all those are the host defense responses. However, if you're able to measure that, it can tell you about exposure. It can tell you if you had been sick with that bug before. Okay, so they are powerful techniques, but they're not always useful for finding out this. Like you're not looking at the bug, you're looking at the response. Okay, test for known organisms. What test did you give for SARS-CoV-2? RT-PCR. Yeah. So it's a nucleic acid based test where you check for the RNA of the virus. For that, you have to know what the virus is. If you don't know what the organism is, those kind of tests are not useful. Okay. So, and then there's this approach, and I'll tell you a little bit more about what I mean by sequencing in the next slide. So in that approach, we look at all the nucleic acid that is present in a sample. We map it to whether it's a virus or not. So going back to, does everyone know what this is? What is it? Okay, so, and what are these ATGC things? So they are the units that make up the DNA. So you can think of DNA as a long stretch of a string in which there are 4 different colored beads, okay? And those beads are these 4 chemicals. And it is, what do you think matters? Do you think that the number of A's that are there, 8, so we're just going to call them by their first alphabets, ATGC, right? What matters? Do you think that the amount of A's that are present, amount of T's that are present matter about the string? So if I tell you that I have a string about 1,000 base pairs long, and I want to know something about it, what is the most useful information? The sequence of the arrangement of the 4 bases. Okay, yeah, so that's why, so that is the most important information we have. And what do you think about like how universal is this DNA? Like, is your DNA different from the DNA of, say, a bacteria? How is it different? Is it different in its composition? Like, does it also have ATGC or does it have something else? Sorry, I need some shout out, huh? Actually, RNA has U instead of T. For the purpose of our conversation, we will consider them interchangeable. AUGC is the same as ATGC. Okay, let's just consider that. So RNA has U and DNA has T, right? So, but the actual DNA or RNA is universal. That is the composition, the chemicals that make it up are universal. So whether I'm looking at a bacterial sequence or I'm looking at a human sequence, I'm looking at the same chemicals. And because of that, the chemistry for sequencing is the same for all organisms. Okay. So this is the sequencer that I use. What happens here is that there are these bunch of nanometer size pores, and the DNA is threaded through them. And as the DNA goes through this pore, you get an electrical signal across this membrane here. And these signals are then converted back into what the bases might be. Okay, so this is the principle of sequencing for this technology called nanopore. The device is actually very small. It's a handheld device. This is how, and this thing is called a flow cell, and all the pores are embedded here. So you have roughly about 2,000 pores embedded in this space. And as DNA goes through it, you measure the current, and that's how you get the sequence. So once you know the sequence, how will you know what organism it is? The base organism for each organism is different, but how will you know which one yours is? Somebody said mapping, I heard that. Map it to some known sequence. Palindromic sequences are usually present in restriction enzymes. So they're very specific functional elements, but they're not like, that will not help you identify an organism. It'll help you, it'll tell you probably this is a restriction site. So it'll help you in other things in molecular biology, but not for figuring out the organism. So do you think that there is some, where would you find, who do you think has sequenced all the organisms and put them anywhere? And why would they do that? So just for like helping humanity, somebody would have sequenced all organisms and put them in a database. Actually, this is true. There is such a database. Yeah, so, but somebody made the choice. of making such a database and making it openly available. So scientists from all over the world deposit data. There are 3 databases and they're synced among each other. So there is one in Europe, one in Japan, and one in the US. The one in the US is NCBI. And all of them get synced with each other. And so everyone can have access to those data. So what you do is you compare your sequence to all the sequences that are present in that database. And so what you're doing is an alignment type matching. And there are a few algorithms to do that. Have you heard of BLAST? Basic local alignment search tool. That's one of the most common algorithms used to do that. Okay. So this sequencer is really small. There are really big sequencers as well. And it used to take a long time and a lot of effort to sequence all genomes. And this is an astronaut sequencing with the nanopore in the International Space Station. This is a researcher sequencing in Antarctica. I have tried to do sequencing in Uttar Pradesh and in Assam. And I can tell you that while the sequencer is supposed to work in all conditions, including outer space, there are spaces that it does not work in. Like it does not work so well with high humidity and high temperatures. I don't know if you can see it here, but there are ice packs around this sequencer trying to get it to sequence. Anyway, so we have sequenced it. We have taken the sequencer into an outbreak location and tried to find out what bugs are circulating there. So we think that it's possible to do this, and we think that this is the future of how we can integrate molecular biology tools in outbreak response. What that will help you do is understand what are the bugs, and sometimes it's a single bug. Sometimes there are multiple pathogens causing the same kind of syndromes. Then you will get to know that. So I'll tell you more about what we did in Assam. But before that, we'll go back to SARS-CoV-2. This is a sort of, this has been a virus that has gone everywhere. And so what do you think this is? Can you see a little bit of what's there? Yeah, it's basis. So this is actually the genome of SARS-CoV-2, okay? So if you have this information, you have the code of how to make this virus. And if you synthesize this end to end and you introduced it into a cell, the expectation is that you will be able to grow that virus. And so this is just showing you the same thing as all the regions that we know and what they code for. And there is this one small region towards the end of the virus, which codes for the spike protein. Okay. And this spike protein is the part that you saw this, right? This is why they're called coronaviruses. They have spike on, they have the spiky outer thing that goes and attaches to the cell. Okay. And so changes to the spike protein, we thought would be unlikely because they would change its interaction with the receptor. That is the thing that it's going to bind in the host cell. And it's also going to be the thing that will restrict which hosts it can infect and so on. But it turned out in the case of SARS-CoV-2 that this was not true. The spike turned out to be, that's why initially, because people thought that the spike would be stable, that's why all the vaccines were designed to this region. But it turned out that it can take a lot of mutations. It's quite a flexible molecule. It's able to tolerate a lot of changes and still perform its function. Okay. So what are mutations? If you look at this genome here, you see a single base change. That is a mutation. If you get a deletion of a bunch of bases, that's also a mutation. And through time, these mutations accumulate. Okay. So we used to think that roughly you get one mutation in 2 weeks. And as the virus has spread throughout the world and has had a lot of people that it could infect, a lot of combinations of this genome have come about. And even early on, this slide is from 2021, I think. We saw a lot of changes. So this is the number of mutations in different positions of the genome. And we could already see that this virus was changing quite a lot. So there needed to be a way to track these changes, right? So one of the ways to track them is to look at all these mutations and try and figure out the relationship between them. Okay, so this is a phylogenetic tree. So you're trying to use this mutation, roughly say that it is mutating with some, at some time interval. So roughly say one mutation in 2 weeks or something like that, and then trace back when you see a certain number of mutations, trace back when two sequences shared their last common ancestor, and try to make them sisters based on that. Okay, so that's, I think I'm explaining that. Okay, that's roughly how you figure out the evolutionary relationship. So sequences that share an ancestor very far back in time would have evolved quite a lot now, right? So they would have more differences than two sequences that share an ancestor more recently in time. However, this gets broken down if the virus circulates a lot and it picks up the same mutations again and again. Okay, that's called convergent evolution, and that also happens with SARS-CoV-2. So at some point, this nice relationship between number of mutations and the distance between two SARS-CoV-2 genomes, it breaks down. But for the purposes of what we are talking about, let's just consider that there is a phylogenetic relationship. If you see mutations, you can infer what the relationship between two sequences is. So this is a nomenclature. You would have heard this, right? B.1, B.1.617 and things like that. So this nomenclature has everyone heard these kind of, or remember? Okay, doesn't matter. So for the purposes of this talk, because let's say that initially the viruses that were circulating in Wuhan were, there were two closely related viruses with a single mutation. So the SARS-CoV-2 genome is roughly 29,903 base pairs. That's not roughly, that's precisely. These two had a single difference, single change. And after that, now I think there are like thousands of base changes in circulating viruses. And this way, it captures what the parent lineage was. So when I call something B.1, I mean that it has a parent in B. If I call something B.1.1, it has a parent in B.1 and in B, right? And about 4 steps down the line, you change the alphabet. So this is just a nomenclature. This was used for SARS-CoV-2. And for what I'm going to discuss next, you just need to know a little bit of this. That's why but they also tried to keep an epidemiological relationship. That is, something was called. So what is called B.1? Does a single mutation make it B.1? So in order to call something a new lineage, in this nomenclature, they said there must be an epidemiological link. That means there must be a cluster of cases, okay, of when you look at, so in this epidemiology thing, we like to know who got infected, when they got infected, and where they got infected. Okay, these are the 3 things we care about. And if there is a relationship there, we call it a cluster, right? If all of them, and then if in that cluster you get the same genomes, that is the same constellation of mutations, it is very likely that it's a lineage. So that's how they define a new lineage in this nomenclature. Initially, when SARS-CoV-2 was sequenced and put in its phylogenetic tree, this is what it looked like. So the SARS-CoV-2 that the new one is here. The old SARS-CoV-1, which was a 2003, I think, right? Yeah, which was a different virus, is quite far away in the phylogenetic tree. And you can see that the closest relatives of this virus that has been circulating for the last 4 years or so is a virus in bats. Okay, this is an early paper. A lot more has been sequenced. We know many more animals that have SARS-CoV-2-like genomes. And but early on, it became clear that this was going to be a distinct lineage. It belonged to the corona. So what did this tell us? What did the sequence, when they sequenced the first SARS-CoV-2 genome, it told us that it was a coronavirus. It helped us put it in this phylogenetic tree and closer to bat viruses. So that's the information that we got from the genome. Now, people have gone back and mapped out all the mutations that are present in many different animals. So this, like I said, this spike of the SARS-CoV-2 is actually quite plastic. It's able to take a lot of mutations. And these are like the different hosts in which those mutations were found. And so the reason why we care about this is that if there is a circulation of a virus in a particular host that humans don't come in contact with, and we develop immunity towards a different variant, if that spills over again, then we may be susceptible again, or vice versa. So that's why we try to keep track of this, right? So people have used genomics to figure out where we think there was an epicenter or the first outbreak of SARS-CoV-2 and where it spread from. So this is a seafood market And this is some initial spatial distribution of the cases. So this is the WHO case from the WHO cases. And these were the cases with known contact in the market, I think. And this was these were all the cases with no linked contact, but you can see spatially they are in the similar region. And this is from an app that's popular in China of the infected cases. And you can see that it's sort of clustering around the market area. So this market was shut down very early on in the pandemic, and they cleaned it out completely. But afterwards, it became important to try and try and understand what happened there. So they sampled, so all the animals had been killed at that point. So there were no live animals. So all of this data that we have is from environmental sampling, wastewater sampling, swabbing of different cages and things like that. And they were able to come up with this one particular side of the market where they could detect more positivity than the rest. So this was by RT-PCR, and they got some animal cages in which they could detect some remnants of the virus. And this is what the data looked like. So basically they had these 718 samples, of which I think 40 were positive by RT-PCR, and then they were able to isolate viruses as well. Right, so, and it was present in drainage, it was present in multiple, but very low positivity, probably because they went in quite late. And so from the sequences that they got from that market, this is the sort of idea we have of the origins of SARS-CoV-2. We can never know for sure because we are trying to reconstruct history, right? So, so we think that there was some kind of reservoir species from which some animals who were intermediate hosts. Now we know that a lot of animals are susceptible. Intermediate hosts probably present in the market got infected, and there was widespread circulation among animals because two lineages evolved, and A and B, and both of them were independently circulating in the market after that and infected human beings. So this is, so basically we have from this here and here, we have two events. That is the common ancestor for the two lineages was probably further back in time. And so this is a phylogenetic tree, and these are the two lineages that were circulating. And there are ancestors here who, that were not sampled, okay? So we didn't get them, we have just reconstructed that they probably existed. So this is the same thing again, so with just more details, that there's probably some reservoir species in the wild in an unknown location, and probably some wild animal, because there was a lot of wild animal trade also in this market. There were some people who had gone to this market years before and taken photographs. So that's how we know about some of the animals that were present there. So there's an unknown, so that animal probably, once it was there in the market in these cages, is where actually these animals also kept very close to each other, cages, in crowded cages and so on. So probably transmission occurred there. So this is one way of looking at that story. And then you get an epicenter at Wuhan, at the Wuhan market, and then in the city of Wuhan, and then global spread. Right, so what do we think happened in Karnataka? So this is the part of the story that I was involved with. So I'll tell you a little bit about how we use genomics to try and understand. So this was very early days, between March and May 2020. First case in Karnataka was detected on March 8th, I think. Yeah, so in that time, in the time of this study, there were roughly 1,500 cases. And we looked at what are the lineages, that is how many different SARS-CoV-2 lineages were there early on in those sort of, in that timeline. And then we asked, how did the virus enter Karnataka? This was, we couldn't answer that fully necessarily. How did it spread and how did it change? So these were the questions that motivated us. At that time, so this is work that was done when I was at NIMHANS. It was one of the centers that was doing the testing. So what you're seeing is a heat map and colored by the number of cases. As you can see, Bangalore city here has a lot of cases. And another cluster was in Mysore, one of the largest clusters. And then you can see in this north, this is where the border to Maharashtra is, right? So yeah, so this was the state roughly at that time. And The ones that were tested in NIMHANS are in dark green. So we didn't get samples from everywhere. We got samples from some of these sites. And the ones that we sequenced are in line. So it tells you we sequenced a fraction of the cases. And at that time, we were getting most of the asymptomatic people. Okay. So this was after the travelers had come in. So this is sort of slightly later. We are getting samples slightly later. And what we found was that there were multiple different lineages of SARS-CoV-2. At that time, we were thinking that the virus doesn't change too much. Okay, that is what allowed us to think that when there is a difference in what we see, it probably came from different places rather than it is changing here itself. Okay, so this was an assumption that is there behind this. Mostly true, but as you can see here, the virus is also changing. So you can see the lineage change within a particular part of this phylogenetic tree. That could be because the virus was already changing after entering Karnataka. So we had a major lineage called B.6, which never took off anywhere else in the world, but was a big lineage in India. And we had some lineages that we never saw again, like B.4 was contained. It was only present in that time. We never saw it again. And the only lineage that took off of all of these was B.1. It was the same lineage that caused the large outbreak in Italy, and all descendants of all the viruses that are circulating are descendants of B.1. That's how successful. There was one mutation that set it apart from all the other viruses, and that gave it such a big advantage that it took over everything else. Right, so out of these 1,500, roughly 1,500 cases that we had, we sequenced 101. And we had the contact history. So this is one of the things that really helped us do a good study, is that we knew who were the travelers. We knew who were domestic travelers, where they had come from. We knew whether they were people who did not have a known history. So we were, and people who were under investigation, but they had no known contacts as well. So we were able to make this sort of cluster. And here we were able to, so each circle here is a person, and if there is a line connecting them, that's because they were known contacts. Okay, and and they are colored by lineage. Everyone that we sequenced is colored, and they are colored by lineage. So what you can see here is in most of the 17 largest clusters that we had, there was a single lineage in them. This, for example, is a cluster in which there are two lineages. They had suspected that this person is the person who infected this cluster, but it turned out to be not true. This was, in fact, a quarantine center. And this person was not in that center, had just been visiting. And this was one of the largest clusters in Karnataka. And so we looked at whether the people who had the maximum number of connections were symptomatic or not. And so basically, these are colored. Asymptomatic people are in sort of this yellow or cream, and symptomatic people are in black. And what you can see is that symptomatic people tend to have more connections. Either they are the spreaders or they are the seeders of the clusters. This is what we thought at that time, but we had very few of these. So we really didn't make that conclusion. From what it looked at that time, most of the clusters seemed to be in a particular district, like one district or another. They hadn't, but there were some of the large clusters had spread to multiple districts. This one. So this is what the situation looked like. And this is what we call the epi curve. We are looking at the number of cases over time. And this is how those clusters were spread out over time. And we could tell that some of the clusters looked like they were, at the end of our study, we could tell that some of the clusters were contained. They were not growing, but some of the clusters looked like they would continue to grow. Right. So this is very little data. So we didn't go into that too much, but we had that. So from this, we could basically say that there wasn't a single introduction. There's not one lineage that came into Karnataka and then spread. There were multiple introductions into the state. There was continued importation with domestic travel, even though there were lockdowns. There continued to be these events of people coming in with the virus. The largest clusters were effectively contained, but there were still clusters that were ongoing. And we could assign lineages to people with no known contacts or travel history. And so we knew which ones were not getting contained. And symptomatic individuals are spread across multiple lineages. So it didn't look like there was a particular mutation that was making people more sick than others at that time. Okay, so that was also a concern. Is there one lineage driving the entire outbreak? And after this, we continued to sequence, and we were able to detect some of the variants of concern as soon as they came in. And the only important thing that I think that we learned from that entire exercise is that a variant can emerge anywhere in the globe, but it spreads really, really quickly. Okay, so within a week to 10 days, you're able to detect it in all major airports. And I think that that's an important thing. What we learned from that is we need to continue having the sequencing type efforts locally, and it needs to continue over time. We need to track viral evolution. It does help to understand what's happening in the local scenario. We missed Delta in a critical window that if we had sequenced more, we could have found it a bit earlier maybe. We need to analyze sequence data together with who is getting infected. Just having the genomes is not enough. And then there was a consortium that was set up to do this kind of sequencing. And post that, we've been trying to look at SARS-CoV-2 in wastewater. That's one of the things that I'm involved with. So what did we learn from this entire thing? I think that we had to build the tools, right? So before this, if you look at the number of sequences that India was contributing to this global database that I told you about for viruses, there would be, how many dengue sequences do you think that we have? If I told you that roughly we have 100,000 SARS-CoV-2 sequences that India has contributed, okay, how many dengue sequences would you estimate that we would have in that database? Give me the order, like 10,000, something. Thousands. Actually, it's in hundreds, and it used to be in the lower hundreds. And for some of our really important viruses, we have like single-digit number of sequences. And the issue with that is that when a virus changes, we are not able to track it. But we have also not been able to make a convincing case that this information will get used. So I think that we have problems on both sides. We don't have the sequences, and people don't know what to do with them once we have them. So if we make a good case for what to do with sequencing data, we can ask for more sequencing to be done for other viruses as well. Right. So when we started in the beginning of this pandemic, we didn't have that much experience with sequencing viruses, okay, throughout the country. We didn't have that much experience. We didn't know how to call the spike gene. So how do you know out of that genome which part belongs to what protein? We didn't know that. So that's called annotation, that you compare it to a known sequence and you figure out what that could be coding for, right? And we didn't know how to do some of that. So we struggle quite a lot. What do you call a mutation? What is your confidence in that? These kind of things. And that comes only when you do something a lot, right? So we had to build the tools. In this pandemic, we got the opportunity to test our ability to do that. We needed to do in a timely manner. So not just do it right, also do it in a 2-week, 3-week window when it will be most useful. It wasn't very useful to give sequencing results a couple of months down the line. It was required within a week to 10 days, if possible. But we were still doing it quite late because we were figuring out how to do it. So in order to build confidence, it's important to keep doing something. And we needed to achieve some scale. For the entire country, having some 10 sequences was not enough for tracing the virus. And for SARS-CoV-2, it was important. And so, there were many mechanisms by which this was done. And sometimes when you scale, you forget what's important. So what happened was we got more interested in increasing the number of the viral sequences in a database rather than asking what is changing, what is its context, is there a new variant? Because it was too hard to ask that question while also doing thousands of sequences at the same time. Okay, so there was a challenge that of losing sight of what is important when you're trying to scale. And this is the place where we are now. Right now, this is not a crisis situation, and it's hard to keep things going when there is no crisis, right? Right, so I'm going to change gears and talk about a little bit about a different outbreak. In an outbreak situation, knowing the pathogen can help you make some decisions. Okay, that's the sort of take home of this part. And we need to do that so that we can bridge the gap between public health and the kind of work that I do, which is laboratory based. In India, we have a lot of outbreaks. And if you want to ever look at that, there is a surveillance network that collects this information. It's called IDSP. It's an integrated disease surveillance program. It works. There are officers in every district, and they collect information on if they are seeing a signal above what they expect. Okay, then they call it an outbreak, and they report the number of cases. We don't usually have molecular investigation. We don't know what is. So they will just report syndromic, like there is an acute diarrheal or there is an outbreak of fever. We won't necessarily know what is causing it. And the infrastructure to do that is still being put in place. But right now they are trying to do this. And so you will get these reports. Usually they will be an image on a website. So in Assam, and we know, so from this, basically that viral acute encephalitis syndrome, while the number of total numbers are small, it's actually a very devastating inflammation of the brain, and usually affects children below 15 years of age. And you get either you have like children dying or you have lifelong disabilities, okay? And it disproportionately affects certain states in India. And JE is Japanese encephalitis. It's Japanese encephalitis virus caused by Japanese encephalitis virus. It breeds in rice fields. The vector for that virus, which is a mosquito, breeds in rice fields. So it's very, very specific to the kind of locations that you will get this virus in. It breeds in rice fields, it amplifies in pigs, and it is transported by migratory birds. Okay, so you have migratory birds introducing this virus every cycle, probably. That's what we think is happening. And you have it biting, like the vector, biting the migratory birds and then carrying it to pigs and then amplifying in pigs. And then the same vector then is biting humans. And so during the post-monsoon, July, around that time, July to October, is seasonal outbreaks of JE. And this doesn't matter. So here you're looking at how cases are climbing in weeks towards July, August, I think. And I think around September, it sort of ends, this graph. And what you're looking at is, JE positivity. So you see high positivity for Japanese encephalitis virus throughout. And you see that the number of cases of acute, that syndromic, that is acute encephalitis syndrome, and Japanese encephalitis virus, the number of cases detected, the trend looks the same. So this looks like an outbreak of Japanese encephalitis virus. So we went there and we set up the sequencer that I told you about in that location. And this is when a sequencing is going well, everything looks bright green on the screen. So this is a good sequencing run. And we got complete genomes of Japanese encephalitis virus. And it where it falls in the phylogenetic tree is in genotype 3, okay? So there are 5 genotypes of Japanese encephalitis virus, and we were worried that there was genotype 5 was the one that was circulating because the vaccine that is currently being used does not cover that genotype. But actually it turns out it was the same genotype as the vaccine strain as well. So it should be covered. So all we need to do is increase vaccine coverage. So that was the so basically we got complete genomes. We were able to see that it was genotype 3, and we were able to suggest that you need to vaccinate more. And so basically, when you combine information on pathogens with who's getting infected, where they are, and when it's happening, you can get insights during an outbreak, and this in turn can inform policy and suggest interventions. Lots of people have helped with this work. I was funded by all these people here and lots of teams. I'm not going to name all of them, but this is a brain infections group that works across countries. This is NIMHANS, some people from NCBS as well, and lots of people during the COVID time. And some of the work that I showed you was done by these people. Farhat helped with making all the cluster that you saw. Yeah, that's it. I'm happy to take questions, have more discussions, but that's the talk. Thank you.
We can open the floor for questions, right? So if anyone has any questions.
Yes.
Good evening. I had a small doubt. You had mentioned in the beginning now airports, shipping docks. Because of that, there's a lot of traveling, a lot of connection. And you had briefly mentioned that even mosquitoes, they enter the containers and they travel. So my first question was, do they actually stay alive long enough to, reach the other side? And
Yeah, so apparently, when these tires get transported, there is water in the tires. Apparently, that's one way that mosquitoes have found a way to go from one country to another. This is known. So it happens.
And when these mosquitoes are traveling, like through shipping containers or just through plain air,
Or people.
or people.
I mean, the viruses
Yeah,
can,
yeah.
but then you need the host in the other end. I mean, you need the vector in the other end also.
So my question is, do these viruses mutate within the mosquito's body as well?
Right. So people have looked at the diversity. So we expect that the selection pressures will be different in a mosquito than in a human. And people have done that work and have found that the sequences are different. They do mutate. All these viruses, every time they make copies of themselves, they mutate. So what happens is they have ways to make copies of themselves, and they don't have good correction mechanisms. So when we make copies of our DNA, we have got some checking mechanisms in place to correct errors. They are more, some of these viruses, particularly RNA viruses, are more error prone. So they will mutate irrespective of the host. Which are the mutations that survive seems to be dependent on the host. So mosquitoes have a slightly different spectrum of the virus than humans.
You had mentioned there was this slide called "How Do Pathogens Emerge," in which we saw like the data for wildlife animals, for domestic animals, and for humans. So for domestic and humans, it was kind of similar: one long spike, then a downfall. But for wildlife, it was kind of like, sorry for the mathematical reference, like a sin X or cos X function.
Yeah.
So is it possible that our body defense mechanism and the wildlife body defense mechanism is different? Like they're used, it's common cold for them.
Yeah, it looks like that. It looks like, and that's when we call something a reservoir species, when it doesn't seem to be making them sick necessarily. And I was trying to think of, it is, for example, every time there is a young in that population, they get the virus, they get an infection, but then they make antibodies. And so now the entire population is again immune. So you get spikes again when there's a susceptible population. That's my best, but you're right. It is that if we call something a reservoir, if they are not getting sick, and people have looked at the immune system, and people study the immune system of bats a lot to try and understand this, because bats seem to harbor a lot of these viruses. We don't know if it has an impact on them. They have a high metabolic rate or something like that. And I think they have a lot of differences in the DNA damage response, but not in the immunity genes. This was the first surprise, but then people are working on this. I mean, I don't know the latest in this field, but this is something of interest. Yeah.
Okay, one last question. For the nanopore sequencing, how do you extract the DNA?
So generally, for all DNA extractions, the idea is that if you put detergent, you take the cell out. So you can take your regular,
like out of blood.
Out of what? So in the case of SARS-CoV-2, we're using nasopharyngeal swabs. In the case of people with brain infections, we'll use their cerebrospinal fluid. So whatever is being used to test for the infection that they have, so blood as well, we are taking out the DNA from that. But the principle is that you break down the cell to get the DNA out, and then you stabilize it, and then you purify it. These are the 3 rough steps.
That's all. Thank you so much.
Thank you.
Good evening, ma'am. In the earlier slides, you had showed that India was one of the hotspots for an outbreak, and most of the other places except for China and India were green and yellow. Why is it that?
So first of all, that's a predictive map, okay, based. So it's a prediction based on what people think are known risk factors. But if you look at it, there are other regions as well. There is parts of Europe that is in red and yellow as well. Yeah, you can see some regions here as well. These are the biggest. So in this map, it is red because of the high biodiversity and possible interaction between wildlife and human beings. But that is one of the hypotheses. Like this is a predictive map, and in principle, a virus could emerge anywhere where you have wildlife interacting with an unexplored region. So in South America as well, people think that if you go and encroach into places where there may be this virus circulating, you can get contact with it. So basically, we're looking at what is your probability of contact with an agent that you haven't seen before. That's what this is based on, but this is just a prediction, like it can be wrong. But yeah, that's high biodiversity, high population, high domestic and small interface between wildlife and humans. Those are the factors.
Thank you, ma'am.
Two questions. First one, we are on this map and I had a question related to this. What happened to the Amazon? Like
it looks a bit, I don't know. I don't know. Because I think if we zoom in, it's just a large area, right? If we zoom in, there are definitely red spots here, for sure. And there are red spots here as well. It's just that this looks so bright red that we are not seeing the other reds very clearly. But there are reds in these places as well. They're just wide areas and small red spots. Yeah, I think we should try and do this map and see. And the other thing is the population is lesser. Yeah. So, I mean, the number of interactions, if you model, it will be lesser. So lesser probability of coming into contact, even if there is, but there have been books written about this, by the way. There is a book, I think, about when you open out the Amazon and go in there and finding viruses that you didn't expect.
Also, the second question was, is there a particular reason why viruses usually, are, where mosquitoes become the vectors for viruses?
Well, firstly, mosquito, okay. There are other vectors. They are also quite common and they do, I'm trying to think. Yeah, some of the flaviviruses have Arctic bone. It just looks like human urbanization has done good things for mosquitoes. And so if we lived in an environment or built environments in which ticks could do very well, we would have, because this has sort of tracked with human urbanization.
Great talk once again. I had a question based on the phylogenetic relationships. Since it looks like a map, is it possible to predict which mutation is more probable to happen? Or like which mutation is more deadly or riskier?
So people do look at that. So when I said, do you remember I said that that particular B.1 had one mutation that sort of took over. So when you see a single mutation and you see a takeover event like that, then you can ask, is this mutation giving a selective advantage to the virus? So that's how you get to know about that question. It's not exactly answering what you asked. Can you predict beforehand what mutation? So people have tried to do that as well. The way you usually do that, it's called gain of function experiment, where you grow the virus in a lab and you let it go through a population and look at all the mutations that come about, and then sort of make a probability map of what are the high likelihood ones. But for that, you have to grow the virus and let it mutate. Can we predict before? You can also make predictions in silico about what are the likely mutations that can happen. And that is how people understood that SARS-CoV-2's spike is quite plastic as well. No, they also did the experiments, both. You can make predictions, but they are just that.
So it's slightly random until you can do experiments.
There are some things that don't change as much as other things. So if something is a structural protein, so if something is the polymerase and it is the active site of that enzyme, that is unlikely to change. But that's the exact idea with which we thought that the spike won't change too much. But apparently it does. And apparently people in the coronavirus field knew about it as well. As in, it was not so surprising to them because they'd seen other coronaviruses change. So if you know a virus family, you can have some idea about what regions will change and what regions won't change. Yeah.
Okay, thank you.
The cluster maps that we saw, how exactly do we go about making those maps and how accurate are they?
So these were made from data that we got from people. And so they are clustered by, did this person have a contact? So this is like, so when you have an outbreak and things like that, people go out and ask for information, right? So did you have contact? And usually, who are the people you had contact with? So this is, in that time in Karnataka, people were doing that exercise. And for every person who was positive, I think about 40, 50 people were traced. And so we could get that. It was only because it was early on, the number of cases were few, and we had these records that we could do it. But when you do have the contact history, when you can tell who is the person they likely had contact with who was positive, it's possible to build these maps. Shiva, do you want to add anything more? Okay. Yeah, but the data is basically, we had a list that told us who, so we had this as P1, P2, P3, and we would know P3 is a contact of P1, P2 is a contact of P1, and so on. And then we would have no contacts and things like that as well. So the ones that you see outside of these clusters are that.
But it seems like this method has a lot of is prone to error. And even the testing, we heard a lot about false results coming out from the test. So how do we know that asymptomatic people were?
We don't know that. We don't know. Okay, so that kind of accuracy. Yeah, all of these are based on what is reported and what is recorded. So whatever errors are there in the record will translate here. And as you can see, we already had, right? This person was put in this cluster, but it does not look like this person belonged in that cluster. So we have so how would you verify that? You would have something orthogonal, some other method for checking this kind of thing. So you, how do you know testing numbers? You look at seropositivity, right? So at the end of the day, if your seropositivity is high, irrespective of your testing numbers, you know that people have been infected. So if they have an immune reaction, they have been exposed. So, but whether they fell sick or not, that you can't tell, right? So,
thank you.
Sorry. You want to say something else? No, the clustering is based on line-list data, not on lineage, because we only, so the cluster is about 1,500 people or something like that, whereas the number of people is sequenced is only 100. So
ma'am, you said that if there's convergent mutation, then it would go back to a similar type of mutant. So would it, if it goes to a similar or the same one as before, would it continue doing random mutations? Or would it somehow have learned that this is ideal for it?
So, so basically you only know that when you apply the selection pressures, right? The idea is that you have some mutation gives you a huge advantage. So even if you had very different origins and very different ancestors, you tend to have that mutation, or you will pick up that mutation as you go along because that gives you an advantage. So we have seen that in SARS-CoV-2. What is the second part of your question?
If it goes back to the same or similar one, then would it continue being random?
It looks like it, because your backbone is now different. So you diverged a long time ago from a common ancestor, but you picked up one mutation, which seems to have an advantage or something. So both lineages now have that mutation, but you still have this backbone of change that has happened in that time, right? So you will continue to have some of those as well. So that's why you have different lineages, but lineages do get completely wiped out and new ones take over. At least for SARS-CoV-2, that is true. For some other viruses, you get co-circulation as well, like multiple lineages. It doesn't fully answer what you said, but because it's not exactly random. Yeah. Every time you pick up a mutation, you also set up constraints. That's why.
Right. So let's thank Dr. Chitra once again for a very enriching talk.